We are delighted to bring you this seminar presented by LabRoots and in collaboration with the National Institute of Health. Welcome to our track on the NIH Brain Initiative, a multidisciplinary approach to neuroscience. We're glad you could join us for this panel presentation titled The Neural Control of Locomotion, an Integrative Approach. It's my pleasure to introduce a panel of experts who will be speaking on the topic of advancing human neuroscience. For a full listing, please click on the tab at the top of your screen. If any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our speakers will respond back to you via email. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, please join me in welcoming our panel of experts.
Hi, my name is Michael Dickinson, and I'm a professor of biology and bioengineering at the California Institute of Technology. I'd like to welcome you to this panel presentation entitled The Neural Control of Locomotion, an Integrative Approach. The locomotion of humans and other animals requires a seamless flow of information from sensory modalities all the way to the motor periphery. As such, locomotion is an excellent system for understanding how the brain modifies neural signals from one processing stage to the next, ultimately transforming sensory signals into a code that controls motor output and behavior. Locomotion is also the means by which animals execute many essential behaviors, such as dispersal, foraging, courtship, and escape. The presentations in this panel represent a sample of the work being conducted by a consortium of 7 laboratories supported by a U19 grant through the Brain Initiative at NIH. Our research exploits a single experimentally tractable model organism, the fruit fly Drosophila melanogaster, in which we can easily study the function of genetically identified cell classes and ethologically relevant behaviors. The talks in this panel, all delivered by graduate students and postdocs, the people who actually do the work, will highlight a range of experimental approaches that collectively constitutes a systematic attack on the neural basis of behavior that integrates vertically across phenomenological tiers. The transect runs roughly from the local motor circuits that control the action of individual limb muscles, through central circuits that regulate patterns of locomotion, to the behavioral strategies that animals employ when they use their locomotor capabilities to search for food. The first talk will be presented by Dr. Tony Azevedo, a postdoctoral researcher in the lab of John Tuthill at the University of Washington. Tony will describe how the genetic tools available in Drosophila make it possible to study the local sensory motor circuits in the ventral nerve cord, a structure that's roughly analogous to the spinal cord of vertebrates. These important local circuits regulate forces generated by individual limb muscles and form the foundation of all behaviors executed via limb motion. The second talk will be presented by Claire Howard, who is an MD PhD student in the lab of Richard Mann at Columbia. Claire will discuss how the neuromodulator serotonin regulates locomotor pattern through its coordinated action on local sensory motor circuits of the ventral nerve cord. This work is an excellent example of how nervous systems use neuromodulation to increase the functionality of locomotor networks. The third talk will be presented by Luke Brezovec, a graduate student in Tom Clendinen's lab at Stanford. Luke will describe new techniques for conducting whole brain imaging in intact behaving flies. These techniques, to which Drosophila is particularly well suited, make it possible to quickly map the function of different brain regions as animals execute specific behaviors. Luke is using this approach to identify regions of the brain that are involved in the visual control of steering. The theme of steering will be continued in the fourth talk, which will be presented by Dr. Sasha Ryshubsky, who recently completed his PhD in Rachel Wilson's lab at Harvard Medical School. Sasha has identified an intriguing pair of descending neurons with inputs in the brain and outputs in the ventral nerve cord. Descending neurons are a critical class of cells because they sit exactly midway in the process of sensory motor transformation. The cell that Sasha has discovered appears to function as a command neuron for steering. The fifth and final talk will be presented by Emily Palmer, who is a second-year graduate student in my lab at Caltech. Emily will describe new behavioral experiments aimed at understanding how animals keep track of their position in space as they walk. By measuring distances traveled in each direction, animals can potentially plot a course back to any salient location in their environment, a form of spatial memory called path integration. Emily will also show how we make use of mathematical models to test predictions of the hypotheses that emerge from our experimental results. So collectively, this series of talks illustrates our team's goal to develop a comprehensive theory of animal behavior that explicitly incorporates neural processes operating across hierarchical levels, from circuits that regulate the action of individual muscles to those that regulate behavioral sequences and decisions. So thanks for logging in to watch. I hope you enjoy the presentations. And the next talk is from Tony Azevedo.
All right, thank you, Michael. All right, so my name is Tony Azevedo, and I work in John Tuthill's laboratory at the University of Washington. And I'm going to tell you today about the neural control of leg movements in flies. And I'd just like to point out at the beginning of the talk that we had help from people in the Tuthill lab, including Evan Dickinson and Pralaksha Gurung. And then we also got started with help from Richard Mann, who's a member of this group, specifically Lalanti Venkat, who helped us find tools and reagents to really get us started. Okay, so the Tuthill lab works on how the nervous system controls walking in flies. And flies are great walkers. They spend a lot of time walking around finding food and mates and places to lay eggs. And they step about 10 times per second. So that's quite fast and requires a lot of coordination of the limbs. And we want to understand how the nervous system controls that pattern of muscle contractions. And as a result, then use the flies as a model for locomotion across animal species. So, but when we got started, we really had no good measurements of basic parameters of leg movements, in particular, how much force is produced by a muscle contraction and which motor neurons are controlling them. So that's what we set out to do initially. So just as background, here's an image of a fly leg. Each leg, 6 legs in all, each leg has 5 segments and 14 muscles controlling positions of those segments. So those segments include the coxa, the trochanter, the femur, and the tibia, and the tarsus, which is not shown here. And then 14 muscles. And each muscle is then composed of several muscle fibers, and each muscle fiber is innervated by a motor neuron, and a motor neuron innervates a group of muscle fibers. So here in red, I'm showing you a stain of the muscle fibers, and you can see several grouped into muscles in the femur here. I'm going to point out the femur in particular. And then here's in green, there's a motor neuron axon that's leaving the nervous system and projecting into the leg. And you can see that this one innervates, has a large axon terminal that innervates several muscle fibers within this muscle in the femur. So this neuron controls contraction in a muscle that results in tibia flexion. So it closes the angle between femur and tibia. And it's one of several that control tibia flexion. So we wanted to understand a very basic thing. How are the neurons that control one direction of movement, in this case tibia flexion, organized to produce different amounts of force and together cause contraction of the tibia? So to do this, we set out to find genetic reagents that label individual motor neurons in the central nervous system that project to the femur. So here I'm showing you what we're calling the fast, the intermediate, and the slow motor neurons that control tibia flexion. So starting on the left up here, the fast motor neuron has a very large axon terminal that grabs onto a lot of muscle fibers. They're all in the tibia flexor muscle. The intermediate neuron projects, this is what I showed you in the last slide, this projects to the more proximal fibers within that same muscle. And then finally, the slow motor neuron, if you're looking just at the leg, projects all the way out to the distal tip of the femur and innervates only 1 or 2 muscle fibers in that region. And then below, I'm showing you what the dendrites in the ventral nervous system look like for the fast, the intermediate, and the slow tibia flexor motor neurons. And you can see that the extent of the dendrites in the ventral nervous system mirror to some extent the patterns of the size of the axon terminals out in the leg. Okay, so we found these tools that label these individual neurons, and then we wanted to understand how they function individually and how they work together to control tibia flexion. So we developed a preparation where we could do electrical recordings from these motor neurons. It's fine. We targeted our electrophysiological pipettes to the cell bodies of these neurons. And at the same time, we then arranged the fly so that it could also pull on a small flexible fiber. And we calibrated that fiber to know how much force was being created if it was deflected a certain amount of a certain distance. At the same time, we then also put a recording pipette into the leg of the fly so that we could measure the spikes in the axons in the fly as well, or in the leg as well. So let me show you what this whole setup looks like together. So here on the top line is the soma recording from this fast motor neuron. And you can see the depolarizations. It's hyperpolarized at first when the fly is not moving, and then depolarizes and fires some action potentials. And then the trace in the middle is this, what we call an electromyogram, this fiber in the leg that's detecting the axon spikes. So you can see that the spikes occur in time locked to the spikes in the soma. And then down at the bottom is the distance measurement of this, of the flexible force probe. And positive direction is flexion. So you can see that when this fast flexor motor neuron fires spikes, that there's big flexion events that we're detecting with the force fiber. Okay, and so then what we can do with this is then cause spikes in this motor neuron. Those spikes travel out to the axon and cause contraction of the muscle fibers. So here on the right, these are time lock spikes aligned at this vertical line. You can see that the spike in the cell body then elicits a spike in the leg, followed by a twitch of the probe. And the probe goes about 50 microns. Turns out that that's about the weight of the fly. The conversion is about 50 microns to about 10 micronewtons, and the weight of the fly is about 10 micronewtons. And we can do this repeatedly. So here are multiple deflections of the same in the same leg. And we can do this for all of the different motor neurons that we found. So here is the intermediate neuron. And these twitches are about 10 times smaller than what we saw in the fast motor neuron. So the fast motor neuron is creating about 10 times as much force as each spike in the intermediate neuron. So finally, the slow motor neurons were quite interesting. They were actually spiking at rest. So you can see here that in the absence of current injection, there's a spontaneous spike rate of about 30 hertz. So at the top here are individual traces with the identified spikes up above. You can see both depolarizing and hyperpolarizing events. Here below that are spike rasters for both depolarization and hyperpolarization. And you can see the hyperpolarization removes the spikes. And the result of that, of the the cessation of spiking is that the fly actually lets go of the probe a little bit. So you can see this negative going trace of the probe distance. So that means that the fly was initially placing force on the probe and then it now let go. Okay, and if we plot the force per spike for these 3 motor neurons, you can see that they cover a huge range of forces. So this is about 3 orders of magnitude on the y-axis, going from 0.1 micronewtons for about 10 extra spikes in the slow motor neuron. Those are the green dots there at the small end of the range. Up to a single spike produces 10 micronewtons, again, about the weight of the fly. Okay, so this tells us something about the organization of the motor neurons controlling flexion in this one joint. In parallel with those findings, we find that each of these motor neurons has a different, it lies along a gradient of electrical excitability. We also find that during spontaneous movements, these motor neurons are recruited from slow to fast in a very orderly recruitment hierarchy. We also find that when we impose movements of the tibia, that there's sensory feedback to these motor neurons that first recruit the slow motor neurons, and then the intermediate neurons at higher perturbations, stronger perturbations of the leg. Okay, so that's what we find in this in our work so far, and this we believe is going to enable us to, in the future, map the inputs to these different motor neurons with EM circuit tracing and improvements in the genetic reagents, labeling either sensory circuits or the premotor circuits onto these motor neurons. We're also going to be able to use these to study the coordinated development of motor neuron electrical properties, the muscles, and premotor circuits. And we can do that with the Man Lab, who has made a lot of tools and made a lot of headway into that research, into that field. And then finally, this ability to measure sensory feedback to motor neurons allows us to start to investigate how those feedback circuits are modulated during behavior, including with neuromodulation, such as serotonergic activation and other things like that, which is something that Claire Howard has been investigating in the Man Lab. So I'll hand it off to her now, and just want to thank you for your time. Okay, thank you.
Hi, thanks so much for a great talk to my colleague. My name is Claire Howard, and I recently graduated from my PhD in Richard Mann's lab at Columbia University. And today I'm going to be telling you about my work trying to understand how locomotion is modulated. So we think of locomotion as a simple behavior, simply just putting one foot in front of the other. But for us, and even for simpler organisms like Drosophila, locomotion is actually a behavior that needs to occur in a complicated and complex environment. My favorite example of this is fast and slow locomotion. We use these 2 different flavors of the same behavior in order to engage with different environmental circumstances. Fast we use to run away from predators, and slow we use in order to engage with our environment and even search for food. So my question fundamentally is kind of how is locomotion modulated in order to produce these different behavioral outputs? The circuitry in Drosophila responsible for locomotion is located in the ventral nerve cord, which is the analog of the mammalian spinal cord. And this structure is composed of 6 hemithoracic neuromeres, each of which contains the circuitry that innervates one of the adult legs of the animal. If we zoom in on this circuitry a little bit, we can see that at its most fundamental level, it consists of motor neurons, which send signals to the periphery to induce muscles to contract. Sensory neurons that send information from the periphery back to the system, and a set of as yet uncharacterized interneurons that comprise the central pattern generator, or circuitry that produces oscillating motor output. So really our question is, how does a circuit like this, that's fundamentally static, produce different outputs to fit different contexts? And this is a question that has interested scientists for a long time, and I want to highlight some work done in the crustacean by Eve Marder and her group at Brandeis University, where they showed that in an isolated central pattern generating motor circuit, application of different substances, what they term neuromodulators, can induce different patterns of motor activity in this circuit. So really, this is kind of the foundation for our work, trying to understand if neuromodulatory substances, for us, the neuromodulator serotonin, are responsible for helping a circuit like the locomotor circuit in Drosophila adjust to different behavioral environments. But before I tell you about some of the results we've gotten in this area, I want to take a step back and think about why Drosophila in particular and collaborations like our U19 Brain Initiative grant are so crucial for trying to understand neuromodulation. What I've told you so far is that a lot of groups have looked at the effect of neuromodulators on circuit dynamics. And this is what originally suggested to us that these substances might be involved in determining different motor outputs. However, what we want to do is we want a system where we can integrate this understanding of circuit level analysis in a behavioral context. And even more than that, what we are interested in is how that behavioral context changes over time in response to sensory information. And Drosophila are really a great model system in order to look at some of these things, because we have great tools to access different circuits, but we also have a variety of behavioral paradigms where we can look at different sensory stimulation. And in a collaboration like this one, when we're working with people who work at the most basic electrophysiological level all the way up to people who are interested in questions of sensory integration, it's a really rich environment to start asking these questions and integrating the data. To give you a sense of how we look at locomotor behavior, I want to share two different assays that we use in the lab in order to understand how animals are walking. One, the flywalker, was developed in our lab, and one, the arena, is something that we brought to the lab in collaboration with Michael Dickinson's lab at Caltech in this Brain Initiative grant. These two different assays are complementary in the types of approaches to locomotion that we can use. So where the arena is low resolution and animals are simply dots on a screen, it provides a wide-angle lens for high-level readouts like speed, angular velocity, interactions between animals, and position within the arena that we can record over long periods of time. In contrast, the flywalker is much more high resolution. As you can see here in this image, this allows us to look at each footprint an individual takes, but it provides a much more narrow focus, and we can look at only short bouts of locomotion as animals walk back and forth across the screen. From this system, we can get footprint by footprint readouts, which allow us to dissect how long the steps are that animals are taking, how coordinated their gaits are, and where their feet are placed as they walk across our arena. And the combination of these two approaches has been really useful in dissecting the role of modulation in locomotion. For example, if we start with our arena context, we can generate a map for each individual of their average locomotion speed across a recording period. And when we activate serotonergic neurons in the ventral nerve cord, we see that animals slow down in response to this stimulus. But this assay doesn't give us an understanding of why animals are slowing down. One hypothesis could be that animals are just so disorganized by activating the serotonergic system that they're not able to access faster speeds because their legs are uncoordinated. This is where the flywalker comes in. And in this system, what we're able to do is map for an individual walking bout each foot placement as the animal walks across the screen, here shown in overlay as if the animal was walking on a treadmill. You can see that every time the animal takes a step, it's very coordinated in where it places its foot. We can also look at coordination by mapping the stance and swing phases for each leg. Here, stance phase, when the animal's leg is on the ground, is mapped in white, and swing phase, when the animal's leg is swinging in the air, is mapped in black. We can do this for all of the animal's legs, and you can see that it forms this nice checkerboard pattern, suggesting that the animal's utilizing a highly coordinated gait. So what happens when we activate serotonergic neurons? Well, it turns out these animals aren't uncoordinated at all. In fact, they're highly coordinated, and you can see that their foot placement remains intact. So it's not that activating serotonergic neurons just totally disrupts these animals. It actually seems like we're putting them in a coordinated state of slow locomotion. But I want to get back to the question that I posed at the beginning of the talk. How do these neuromodulatory systems act to help animals adjust to different behavioral contexts? And in order to do that, we had to introduce sensory stimulation to our assay. And we did that using a vibration paradigm, where we had animals walking in the arena that would experience a sharp vibration stimulus, which is startling to these animals. Control animals respond to this with a particular set of behavioral adjustments. First, they slow down. Then they have a pause phase of their behavior, and then they rapidly accelerate in what we think of as an escape phase. However, when we prevent serotonergic neurons from being recruited into this system, we see that the pattern of this behavior is dramatically different. Particularly, the pause phase, while still present, is dramatically shorter. At the moment, we don't really understand what these different phases of behavior are used for in determining animals' responses to these stimuli. But one hypothesis that we have is the pause phase is important for animals to gather additional sensory information before making behavioral decisions. So our next question is, how is serotonin doing this at the level of locomotor circuitry? And the first important thing to understand is that neuromodulators like serotonin act via a family of different receptors, each of which has distinct effects on the downstream cell. In Drosophila, in serotonin, these families are 5-HT1, 5-HT7, and 5-HT2. And what we find is that, consistent with the fact that these receptors play different roles in altering neuronal cell activity, different receptors play different roles in modulating serotonergic-dependent behaviors, like responses to vibration stimuli. For example, when we delete 5-HT7, we see a very similar phenotype to when we delete the ability of the fly to secrete serotonin. However, when we do a different experiment and delete 5-HT1A, we see a different phenotype entirely. While the pause phase here is maintained, animals reach a much slower ultimate speed in their escape behavior. And this suggests that different receptors, which have different effects on downstream cells, are also mediating different components of serotonin's action on locomotor circuitry. To better understand this, we next ask the question, where are these particular receptors expressed in sensory motor circuits? And not surprisingly, we found that different receptor types are expressed in different components of the locomotor circuitry. For example, 5-HT7 is expressed primarily in distal mechanosensory neurons, proprioceptive neurons, and motor neurons. This is particularly interesting to us because work from our collaborators, particularly the Tuttle Lab, has shown that proprioceptive organs are important for conferring vibration sensation to the ventral nerve cord and may therefore be particularly relevant to the behavioral analysis that we're doing here. Conversely, 5-HT1A receptors are expressed in a different population altogether, primarily more proximal mechanosensory neurons. If we coordinate this information with knowledge that already exists in the literature about what the role of these different receptors are on downstream signaling modalities, we can start building a model of how serotonin could be altering sensory motor coupling in order to change behavior. For example, 5-HT7 stimulates cyclic AMP, which facilitates activity of downstream neurons, whereas 5-HT1A does the opposite. So it may be that activating these different receptor types changes the weight of sensory motor processing, focusing it more on distal and proprioceptive organs. So getting back to the original goal of our work, to start building an integrated model of modulation, I've told you how our work suggests that activation of serotonergic systems induces shifts in sensory motor processing, both sensory and motor shifts. And we think that this is a really exciting area of future work when we can start testing these hypotheses, particularly with the assistance of the Tuttle Lab and their electrophysiological models. Additionally, I've shown you that these systems impact behavior, including slowing animals down and altering how they respond to sensory stimuli. I think the next step is to try and link these two different systems and understand how the effects at the circuit level are ultimately being read out as behavior. And the way we'd like to do that is by inactivating particular receptor subtypes in different subcomponents of locomotor circuitry and seeing how this ultimately affects our behavioral response. With that, I'd like to thank all the people who were instrumental in doing this work, including my advisor, Richard Mann, collaborators here at Columbia and elsewhere, and also highlighted in red other members of the U19 community who've been really helpful as we've done this work. And now I want to hand it off to my colleague. Thank you so much for listening.
Thank you, Claire. Hello, everyone. My name is Luke Brezvik, and I'm a PhD student in Tom Clendenon's lab at Stanford University. And today I'm going to tell you about my work on whole brain imaging of the visual motor transformation. Animals use visual information to guide their behavior across a variety of contexts. One such example is goal-directed navigation, where an animal must use visual and other sensory information to navigate to some goal. In all cases, visual information first enters the eye, and visual circuits extract behaviorally relevant features. These are features such as the position of objects, but also things like optic flow, which is the coherent movement of the visual world as the animal itself moves. These features then inform motor circuits that direct behavior. The degree to which these visual circuits are directly coupled to motor circuits is largely unknown, but examples exist both for very direct connections, as well as more indirect connections through things like navigation circuits. And this transformation is not simply feed-forward, but feedback loops exist at every level. For example, motor signals are relayed back to visual circuits, and the animal's physical behavior itself affects visual input, as in the case of optic flow. Overall, these interactions are a dynamic two-way street. Visual information can guide behavior, but the behavior will in turn affect the visual input upon the retina. The picture that emerges is complex, with all circuits interacting through nested loops across multiple timescales and in a context or behavioral state dependent manner. Given this interconnectivity and coordination across multiple circuits, I think it is fair to say that we will never fully understand these transformations if we only look at each part in isolation. And that is the motivation that informs both my project as well as the overarching goals of our U19 group. To begin to understand these complex networks, I'm studying the fruit fly Drosophila, whose brain, despite its relative simplicity, is still capable of performing many of the same fundamental computations as vertebrates. In the case of visually guided behaviors, we know that first, visual circuits must extract behaviorally relevant features. In vertebrates, as well as in flies, two distinct visual features that are well known to guide locomotion are landmarks and optic flow. Visual landmarks are useful for correct estimation of heading and spatial location, while optic flow helps the animal estimate its speed and distance to objects. This is an image of a Drosophila brain. And on either side are the optic lobes, where early visual processing takes place. We have a particularly detailed knowledge of these early visual steps in processing, and the field is now continuing to get a better sense of how these simple visual features are relayed into the central brain through these projection neurons shown in various colors. For my project, I'm using 2-photon imaging to record neural activity throughout the entire brain simultaneously. When I started this project, Kevin Mann, a postdoc in the lab, had just published the first work of whole brain imaging in adult Drosophila. In his paper, he establishes the whole brain imaging methodology in flies, including a technique for registering individual fly brains to each other and to a common anatomical atlas. This allows quantitative comparisons between individuals and between brain regions. In order to address the questions I'm interested in, the first thing I had to do was implement several technical modifications to this methodology. Namely, the fly must be able to walk during imaging, and we would like to show it visual stimuli during this process. In my setup, I'm imaging from the posterior part of the head while maintaining full optical access to the brain. This is the central brain, and these are the two optic lobes on either side. In this setup, the fly is walking on an air-suspended ball while visual stimuli are presented on screens surrounding the fly. To measure neural activity, I'm expressing the calcium indicator GCAMP in all neurons in the brain. And our resonant scanning microscope allows me to image the entire brain volume twice a second at cellular resolution. I will now show you an overview of the experimental pipeline I've developed. GCAMP neural activity is measured simultaneously with the TD tomato structural marker, which is used for motion correction and brain registration between individuals. Visual stimuli can be presented using software developed in-house with the help of an electrical engineering PhD student, Stephen Herbst. The fly's walking trajectory is measured using FICTRAC software, which tracks the movement of the ball. At the end of each experiment, a high-resolution scan is taken and later used to align individual brains to each other. I've been working on developing new models to extract the brain signals and interpret them as they relate to visual stimuli and behavior. Next, I will show you the results of one of those models. Let's focus on the question, what regions of the Drosophila brain contain information that can be used to predict the forward and rotational velocity of the fly? These two behaviors, moving forward and turning, can potentially be thought of as the two core knobs the animal has access to for directing its navigation. Without getting too into the details of the analysis, let me just say for those interested that I used principal components analysis followed by a regularized and cross-validated linear model using the principal components as input features and the fly's walking velocity as the output prediction. So here, I'm showing you a single slice of the fly brain with each voxel colored based on how well it can predict the fly's forward and rotational velocity. Using this method, we identify many regions already known to have some role in motor control, such as the protocerebral bridge, layers of the fan-shaped body, and regions containing high densities of descending motor neurons. But we also identify many new regions that we are excited to explore. Using this model, we can accurately predict the velocity of the fly given the neural data. Here in black, I'm plotting the true velocity of the fly, and the red is the model's prediction. The top graph is the forward velocity, and the bottom is rotational velocity. This is over a period of 4 minutes. As you can see, the model matches the true data reasonably well, lending support to the validity of the model. The final bit of data I'd like to show you is the same analysis, except now I'm showing you one of the optic lobes of the fly. And despite this particular fly walking in complete darkness, there's clearly an abundance of motor-related signals present. The spatial extent of these signals is much more dramatic than was previously expected. In particular, there are signals present in layers of the medulla, which would constitute the earliest visual circuit neuropil containing self-motion signals. Now, why would there be extensive motor signals in the earliest sensory circuits whose job it is to process visual information? As I mentioned in the introduction, self-motion of an animal will change the visual input the animal experiences. So it could certainly benefit these early circuits to know what visual experiences they might expect and adjust their computations appropriately. For example, if the animal is moving quickly, these circuits may want to be in a mode optimized to efficiently extract information from quickly changing visual scenes. So what I've shown you is I can image the whole brain of a fly during walking and visual stimulation, and that behavioral signals can be extracted using PCA and linear models. My next steps will be to present a suite of visual stimuli while recording walking behavior, continue to map the visual motor transformation across the entire brain, and ultimately identify the neurons' underlying steps in the transformation. Finally, I would like to thank my mentor, Tom Clendenin, and the Clendenin Lab, as well as the U19 project group, whose interdisciplinary collaboration has made this complex project possible. In particular, insights on the central complex from the Dickinson and Wilson Lab has helped me conceptualize and design my project. The Dickinson Lab has led the design and manufacture of the foam balls and ball holder we use to track the fly's locomotion. And the Wilson Lab has helped us get the FICTRAC software for tracking fly locomotion up and running. The Druckmann Lab has helped us develop and explore various flavors of models to relate neural activity to the visual and motor signals. I look forward to our continued collaborations, especially the insights coming from the Wilson, Tuthill, and Mann labs about the logic of the descending motor neurons, which contains many implications to my work. I also look forward to the technological development in the Wilson Lab for tracking individual legs of the fly as it walks on the ball, which could give us a more accurate understanding of the fly's locomotion. In collaboration with the Druckmann Lab promises to yield many more interesting and informative models. And I thank you for your attention. And now I'd like to hand it off to my colleague, Sasha.
Thank you for that introduction. Imagine you need to get to the Washington Monument from Chinatown in Washington, D.C. At first, you'll navigate towards your goal based on a map stored in to memory, either in your head or on your phone. But once you see the Washington Monument, you no longer need this map, and you can simply head towards this landmark by using vision. Steering behavior is an integral part of navigation, and it's a window into the algorithms that the brain uses to guide the animal towards different goals. This example shows that sometimes navigation is driven by goals in working to memory when goals are obscure or out of sight, and sometimes it's driven by sensory cues immediately perceptible in the environment. How the brain integrates these inputs to control steering remains a major open question. But first, we need to answer, where do these steering signals converge? We address this question by studying cells at an information bottleneck, precisely cells in the neck of the fly. There are 100,000 neurons in the fly's brain and roughly 10,000 in the spinal cord, and only 350 descending neurons that carry information from the brain to the spinal cord. And in this talk, I'll show you data from one of these cells. We focus on a cell called A2 because its anatomy suggests a role in the control of walking behavior. And this cell gets its inputs from a premotor region called the lateral accessory lobe that is thought to be involved in forming motor commands for the spinal cord in other insects and also in flies. We're performing whole cell recordings from flies walking on a floating ball, and we use the rotational velocity of this ball to measure flies' turning behavior. This data shows the fly turning. 0 is no turning, and changes above 0 is right turning, and below left turning. I'm making a simultaneous recording of the same A2 cell from the left and the right side. And here's an example trace. The middle trace shows activity in the right cell and the bottom trace in the left. And you'll notice that these cells are generally anti-correlated. Just before right turns, there's a burst of activity in the right cell, and just before left turns, there's a burst of activity in the left cell. If we plot the activity of the left versus the right A2 cell as a function of steering, red is right turning, blue is left turning. When the left cell is more active than the right, the fly is more likely to be turning left, and vice versa. And in fact, we can read off the direction and the magnitude of turn by looking at the difference of activity between both of these cells. We can also derive turns by asymmetrically activating these neurons. I've shown you that spontaneous turning converges onto A2 neurons, and now I'll show you that external sensory drives also converge onto these same cells. We discovered that these neurons integrate multiple sensory drives, and they integrate both attractive and aversive cues. But first, I have to tell you that flies sense odors with their and heat by using their antennae. If we present lateralized odor to the fly turns towards that odor. And if we present lateralized heat, the fly will turn away from that heat. But in both of these cases, activity of A2 predicts the direction of the fly's turn. This tells us two important things. First, A2 is downstream from both of these sensory drives. And second, it's involved in both approach and avoidance behavior. I've shown you spontaneous and sensory evoked turning, and now I'll show you turning based on an internal spatial goal. There's a region called the central complex in the fly that contains maps of space. It also encodes spatial goals. And maps of space are useless if you can't use them to guide turning. And we wondered if circuits in the central complex are used to guide turns via this A2 descending neuron. Having a compass is fundamental to navigation. It turns out that the fly has a built-in compass that is located in the circular structure in the fly's central complex. Strangely, it physically looks like an actual compass. Adjacent cells in this structure encode adjacent angles of the heading, and these arrows in gray represent where the fly thinks it's going. And there's a bump of activity that moves around in a circle as the fly turns, essentially working as a compass needle. To examine A2's role in central complex evoked turning, we first had to inject a turning signal into the central complex. And let me just give you some intuition for these experiments. So your goal is to walk north, and your compass reads north, but someone to direct your compass to point east, and for a moment you'll feel lost and want to correct your course by turning towards the north. We can do something like that with flies. We can monitor the position of the bump by using calcium imaging, and when we artificially set it to a different direction, flies will then counter turn to bring the bump back to its initial position. And it's these turns that we call central complex evoked turns. And we'd like to monitor the activity of A2 during these turns. Here's an example trial. The circular structure that I just showed you, it's unfolded on the y-axis, and the red line is the position of the bump over time. The bump is initially in this location, and we cause a bump jump around this time, and the bump stays at this new location until this time when it returns back to its initial position. We typically see a large turn that precedes this bump return, and this turn is preceded by a burst of activity in the A2 cell. And this effect has been consistent across trials and flies. In the future, to understand how competing steering signals are resolved at the level of A2 neurons, we will need to identify inputs onto these cells. And I've been able to make progress in this direction with the help of the full fly brain electron microscopy data. And using this data, we found these A2 descending neurons, and we identified hundreds of their inputs in the brain. For example, we've identified neurons that directly connect the compass system with A2 steering descending neurons. To summarize, how sensory cues and internal cognitive drives are integrated to guide motor commands is a major open question. And we made progress towards this question by discovering neurons that influence steering during walking. Aversive and appetitive sensory drives converge onto these same cells, and steering drives based on an internal goal also converges onto these same neurons. So these steering neurons are an important starting point for future studies that will examine how conflicts between external and internal drives are resolved. This project has a number of connections to other members of the Brain Initiative team. The Tuthill and the Mann labs are investigating the targets of steering descending neurons in the ventral nerve cord. The Clendenin lab is investigating the representation of steering signals in the visual system. The Dickinson lab is investigating the compass system in the central complex and its connection to steering behavior. And the Druckmann lab is using computational modeling to explore the potential network dynamics of compass steering in interactions. And our next speaker, Emily Parker, will tell you about a behavior called path integration. We think this behavior is driven by the central complex, which connects to the steering descending neurons I have just talked about, and thereby engages the motor circuits in the VNC.
Thanks, Sasha. My name's Emily Palmer, and I'm a second-year grad student in the Dickinson lab, and I'll be talking today about a model of idiothetic path integration. In the 1970s and 80s, a series of experiments were done with the desert ant cataglyphis that studied their long foraging excursions away from their nest. So on these outbound excursions, the ants would walk tens of meters or even hundreds of meters away from their nest until they encountered a food site in nature. And at that point, they would enter the second phase of their search, when they would turn and walk directly back to their nest from that food site. And so this told researchers that the ants were performing a behavior called path integration, or dead reckoning, where they had an internal map stored of their position relative to their nest or their home site. This behavior is really interesting because it allows them to walk directly back to the nest rather than retracing their steps on their outbound trajectory. Once the ants got back to where they believed the nest should be, they entered the third phase of their search, called local search. And in this stage, they would search in the area around where they believed the nest should be, spending the most time immediately around their nest opening and less time further away. And it turns out that not only do these virtuosic desert ants perform local search, but we also see it in flies. Vincent Tatier in particular found that flies will perform local search around a food site in nature, trying to capitalize on the patchiness of food, where they'll loop out from a food site looking for other food sites before looping back to it. More recently, Roman Korfas, a postdoc in the Dickinson lab, found that he could elicit local search with optogenetic activation of sugar receptors in the fly's brain. In Roman's experiments, he had them walk around in a circular arena. And when they entered that little gray dot in the center of the arena, a red light would come on and give them that sugar reward. And from here on out, I'll refer to that zone as the activation zone, or the food site for convenience. Now, before the activation was turned on, before they entered that gray dot, the flies wouldn't exhibit any preference for any area in the arena. They would kind of walk around the walls, sometimes cross the center of the arena. But after the activation turned on, we saw a very different behavior, where now the flies would stay very close to that fictive food site, going on local searches where they loop away from the food and then come back to it. The problem with these dances or local searches is that they're so tortuous that they're very hard to analyze. To start to unravel these behaviors, Amir Babahani, another postdoc in the lab, constrained the flies to circular channels rather than these open circular arenas. Now the flies are allowed to walk in the circle for a few minutes before the activation is enabled. And once the activation is enabled, when they enter the activation site at the south end of the channel, the red light is again turned on and they get the sugar reward. Now, before that is turned on, as in the open arena case, we see no preference for any region of the arena. And they just walk around in circles, sometimes reversing, but tending to just walk in one direction indefinitely. Once the activation is enabled, however, we see the same behavior that we saw in 2D, but now in a 1D case, where they'll stay very close to the activation site and then occasionally make excursions away from it looking for other activation sites. We became really interested, however, in the behavior after the activation is disabled, which spoke to us about what happened when the fly walks away from the food site and comes back and the food is no longer there. To analyze these cases, we termed the distance between the food and the first turn R0 and the distance between those two consecutive turns R1, where there's no food stimulus between those two turns. When we plot R0 and R1 against each other, where each dot on this plot is a different fly, we find a very linear relationship here. And this confirmed to us that the flies were performing path integration, because it meant that the flies were keeping track of how far away from the food they walked before coming back to in the other direction. We fit a relationship to this data and found what we expected, which was this linear relationship. And now this became a path integration problem, where the R1 distance, that second run, is equal to the integrated distance from the food to the first turn, plus some constancy. So we decompose that second run length into these two components. Now, if we assume that local search aims to take advantage of the patchiness of food in nature, it follows that the algorithm that they're using should be able to handle finding two food sites in their environment. And so that's what we look to investigate next. In these cases, when there are two activation sites, the fly first will search around that first activation site. And then at some point, because of the variability in the first excursion distance, it comes across the second. And once it finds the second, rather than just switching to dancing over that new food site, the fly searches between the two. And what was really interesting to us here was that the outbound distance away from the food is shorter on average than the distance between the foods, which means that their algorithm somehow is able to keep track of which direction they're walking in and ensure that they cover both of the food sites. So in the one food case, again, we termed R0 the distance between the food and the first turn. Now we term R0 the distance between the first food that they encounter in that first turn. So this is effectively saying that this is still a path integration problem, but now we're going to be integrating from the first food that we found. So that's when our step counter is enabled. And when we plot this data, we not only find the same linear relationship, but we find the same C value as well, which told us that even though there was a second food found, the flies weren't undergoing any sort of state change. They were using the same algorithm to handle both the search in the one food case and the two food case. Using our integrated relationship, we now have this parsimonious model of how flies are path integrating to perform local search. So they go outbound from their first food. At some point, they execute a turn. And at this reversal, they choose their next run length based on the integrated distance from the food to that turn, plus some constant C. On that trajectory, they come across another food, and that resets their counter. At the reversal, they choose their next run length, and so on. But at some point, because of the variability in their initial run length, they can find the second food. And when this happens at that next reversal, because their integrator started at that first food site, they're ensured that their next run will pass both of those foods. This happens in both directions and proceeds indefinitely for the rest of the search. We implemented this model using a state transition diagram, which is a tool commonly used in computer science for things like autonomous driving, where we have discrete states that the actor can take on, like stopped or walking, connected by probabilistic or deterministic transitions. And this model gives us very realistic traces, very similar to the data that we collected. So you see that it not only works in the one food case before the simulated fly finds the second food, but it also can handle when the fly or the fictive fly has knowledge of both of the food sites. We can also tell the model that there are more foods than this in different geometries, and each of these makes a prediction about how a fly will behave in a new or untested geometry. So for example, the top trace has 3 evenly spaced foods. We can test a real fly in that same configuration, and both the model and the real fly should perform identically. Moving forward, we're really interested in finding out how flies execute this on a neural basis. So what is the underlying neural circuitry governing path integration? And luckily, because we're part of this consortium, we can look to the other labs who are also involved in this project. So for example, the Clandinin lab is looking at whole brain imaging. So we could have a tethered fly prep where a fly is walking on a ball in a head fixed tether. And if we image the whole brain, that could tell us information about which regions of the brain are involved in the path integration process. The Tuthill lab and the MAN labs are working on proprioception and mechanoreception. And since path integration requires that the flies are able to integrate, they must be counting their steps away from the food. And so if we inhibit or silence proprioceptive or mechanoreceptors in the flies, they should no longer be able to count how far they're walking. The Wilson lab is working on steering and compass systems in the fly's brain. If we inhibit or silence the fly's ability to steer, they should no longer be able to navigate back to the food site. So we might see a reduced ability for them to reverse and walk back towards the food. And all of these changes are also implementable in our model. So again, with that example of silencing the ability to steer, if we turned off the ability of the model to reverse, we should see the model and the flies perform identically. Thanks.
Thank you for that outstanding presentation. For additional panels on the NIH Brain Initiative, view our agenda on our events page. As a final reminder, the panelists will follow up with any questions you've submitted via email. Thank you again for your participation.